Hi, I'm Andrew Salywon from the Desert Botanical Garden. Today we've been surveying for the Canelo Hills Lady's Tresses orchid. This is an endangered orchid that's only known from two cienagas, which are freshwater wetlands from the southwestern United States. So the neat thing about this orchid, it's kind of the indicator of the habitat quality. So if you have a lot of orchids, then you can assume that you have a really good high quality habitat. And when those populations are reduced or extirpated or totally gone altogether, then you can assume that there have been either ecological or hydrological modifications to the habitat that have degraded it. Cienagas are important not only for threatened and endangered plants and animals, but for other wildlife in the surrounding arid habitats. So wherever there's water, there's life, and in periods of drought, cienagas act as oases to draw in animals from far away to the water and the green vegetation that exists there because of that water.
Hi, I'm Steve Blackwell from Desert Botanical Garden. I am the Conservation Collections Manager. I manage our collection of rare and endangered species and our seed bank. One of the species that we're working on down here in southern Arizona is the Canelo Hills Lady's Tresses orchid. It is an endangered species that we've been working with the past couple of years. We've actually been working with dog trainers to try to help us locate new populations of this species, as well as locate historical populations that were once known to occur. So here we have a couple of plants, and one of the reasons that we're down here now is actually to collect seeds from this plant. So we've made a little bit of progress from where we were last year. We've actually worked with the Smithsonian, who has been able to isolate the mycorrhizal fungi that go with this species, and they have since sent us samples of that fungus that we are able to culture, and we can use that fungus to grow these seeds in soil mix rather than tissue culture. So a couple of years ago, we've worked out the protocols to grow some of these plants out by seed in vitro or in test tubes using a formula that's specialized for orchids. And we've had some success doing that. And the intention with that is to actually grow these plants out for additional seed collection and also potential restoration out in restored habitats. So now that we've figured that out, what we want to do is try to propagate these seeds using the fungus that occur with them naturally. And so we've worked with the Smithsonian, who has cultured the fungus, identified it, and sent us a sample of it. We can actually use that fungus to inoculate the seeds to get them to germinate and grow out. Typically when you collect seeds or fruits, is you want to get them at their dispersal stage. You can actually collect these when they're slightly still green, and the seeds inside will be viable and actually still sterile inside, so you can sterilize the fruit itself and then save yourself one step in the tissue culture lab. So you can see these flowers already done, and these fruits are all forming along the inflorescence there. And you can see why they call it spiranthes, is because the inflorescence actually spirals around.
Hi, I'm Lauralea Oliver of Canine Incentive LLC, and I'm the trainer for the Spiranthes Dog Project. This is Muon. She's one of our scent dogs. We have two dogs trained on this project, so we have Muon and then Cersei. She's a black Labrador. Muon is a Belgian Shepherd cross. This is our second year doing surveys for Spiranthes. The girls were out here last year and assisting with the surveys then. We had trained them on a different species of Spiranthes back in my training facility in Acton, California. So this last year we've been spending keeping the girls fit, keeping them field ready, doing their maintenance training. I had enough plants that I was able to keep some viable in my screen house back at home. And they went dormant like they normally do over the winter, and they actually came back. I was really happy about that. So I had about 5 plants that came back. So we're able to use those plants to continue their maintenance training. And what maintenance training is when the dogs aren't currently surveying, we have to do things, set up scent problems for them to continue their, understanding of the odor, keep it important and kind of keep it on their radar. So we've been doing that throughout the year to keep them ready. And then as we get closer to the survey time, we kind of ramp that up and we increase the sessions just to make sure the dogs are really keyed in and ready to get out into the field and work. So for the most part, I work my dogs off leash. Occasionally I will work them on a long line if the habitat dictates it. For one of the areas, because it's so deeply vegetated, the plants can be a little bit clustered. I kind of want to avoid walking in the territory too much. I don't want to disturb it all that much with my feet. The dogs are definitely less invasive than we are. They tend to trample things less. It's better to kind of stay out of their way. These plants can take a while to detail for the dogs to really source well, so I kind of like to stay out of their way and let them do their work. So we do a lot of our work off leash. My dogs are really comfortable doing that because a lot of their training is done that way. But we can actually, as a handler, use our bodies to sort of indicate where we want the dogs to work or focus their efforts. So if we have an area that looks promising, I can actually stay in that area as a handler. My dog will work sort of in a circular pattern around me, and I can kind of stick around there and ask them to detail, to really work that area with a fine-tooth comb. And I can use directionals. I can send the dog here, I can send them there, ask them to search. As you can see, she's following my hand right now. So I can sort of ask them to get into areas. And then if I want to move on, if the dog tells me, hey, I checked everything, either they made some finds or they cleared it, we can move to the next area. So I mostly work like that. For one site, we did use long lines because it was a little more open territory. We had a lot more to cover. So my teammate, who's Heather Long, she's my training associate, wonderful. This is her first time out here and she's great, doing a great job. And she's handling the Labrador. So she and I did a pattern where we were both on long lines and sort of traversing across the territory with the wind to our back, just to kind of detail a little bit more and work the area a little bit slower because it was so open. The dogs tend to go a little bit too fast. And when we're looking for something so small like this and so cryptic, we want to slow the dogs down a little bit to increase their rate of detection.
I'm Melissa McCormick, and I'm a senior scientist with the Smithsonian Environmental Research Center and also one of the founding members of the North American Orchid Conservation Center. So the purpose of this project was to identify the conditions that particular orchids needed to grow. And in particular, we were focused on the Canelo Hills Ladies Tresses, which is a particularly rare orchid that occurs here in Arizona. It occurs nowhere else in the world that we know of. And so to understand what conditions it needs to grow, we have been putting loggers out along with sensors, so something to identify the soil moisture conditions that are needed by this orchid, but also to understand the fungi that this orchid needs. So all orchids need particular fungi in order to grow, and we have now, from the work that we did last year, identified the fungi that the Canelo Hills Ladies Tresses needs in order to grow, and then we're using that information now along with the logger information, the soil moisture and temperature data that we collected. We've been collecting since last year and that we retrieved this year. We're using that to identify the conditions that the fungus needs. So all orchids need particular fungi to grow from seeds. These are seeds are very, very small that have just an embryo in them with little or no nutrient reserves. So they need to have a particular fungus to grow from that stage. But even once they become mature plants that produce green photosynthetic leaves, they still depend very heavily on their fungi, particularly to survive stressful conditions. So if they get shaded by, say, vegetation that's very dense all over top of them, and so they're not getting very much light, or if they encounter drought conditions, then they really need their fungi. They can actually shut down their stomates and go back to using just their fungus in order to get the nutrients they need, including carbon. And so then this association with fungi helps them to survive these stressful conditions. And so that's why the fungus is really important, even once the orchid becomes mature. We can grow them under these specialized conditions in the lab without a fungus. And that's often the place that people start with orchid growing. But in order for them to survive in a natural environment and to tolerate the changes in the environmental conditions, they really need that fungus there for them. So this trip, we collected samples of soils in places where the orchid grows and places where it doesn't, places where it used to but is no longer found. And we're trying to identify whether or not the fungus is still there, how much of the fungus is there, and what conditions that fungus needs in order to grow. That then can allow us to determine whether there are places that this orchid might be reintroduced, or whether there are things that we maybe need to change to make the conditions better so that the orchid can grow there again.
Hi, my name is Luis Romero. I'm the Conservation Collections Research Assistant here at the Desert Botanical Gardens, and I help assist with the tissue culture work here at the garden. Here at the Desert Botanical Gardens, we're working with orchids, specifically Spiranthes deltescens. We're growing them in vitro, which just means growing them inside jars. The reason for that being that in the wild, these orchids require fungus and other microorganisms to thrive and grow and flower and seed. Here in the lab, we can simulate that by providing the nutrients in a medium broth. Once we get the orchids from the field with the seeds, we sterilize the seeds, grow them in the jars, and from there we can use it for our research purposes, such as studying the fungus that we're working alongside the Smithsonian with. Or we can even use those orchids for some of our reintroduction efforts all around Arizona. So some of the issues you can have with tissue culture are things from seeds not being viable enough to maybe over-sterilization during that seed sterilization process, or just even contamination. Since you have to transfer these orchids constantly to new media or outside of their media vessel, there's room for contamination, which can lead to the death of the plant. So sadly, all that work you do for months and months can be easily squashed with a simple mistake. So the difference between plants in the wild and plants that we tissue culture is that fungus. Now, some of these plants can continue to grow and even at times bloom, but they won't be anywhere as viable or as vigorous as those with the fungus. Part of the research we want to do here at the garden is to investigate further those fungal interactions and see if the method of propagating these via tissue culture, introducing the fungus into the soil and taking care of them, will help with better reintroduction efforts for conservation.
Hi, I'm Hope Brooks, and I'm a technician at the Smithsonian Environmental Research Center and also with the North American Orchid Conservation Center. And I'm here as part of this effort to restore and understand what's going on with the Canelo Hills Ladies Tresses orchid. A big portion of what I do is help cultivate the fungus in the lab and manage a lot of the small details of the project while everybody gets this wonderful chance to go out and search for this orchid, to work with the dogs, and also to cultivate the orchid out at the Desert Botanic Garden. And so one of the things that really draws me to this project is, first of all, a passion for the desert. The saguaro cactus was actually the thing that got me into botany, and it serves as a great gateway to orchids and other charismatic flora. And so the Spiranthes, which is the Canelo Hills Ladies Tresses orchid that we're studying, is really small and kind of nondescript among orchids, but this tiny white spiral of flowers is also really majestic in and of itself. And it's something that I think is really worth preserving and having more people study and search for whenever they can get out into these beautiful green areas in the desert.
30 years ago, there were 5 populations of the orchid. But in recent surveys that we've done the last couple of years, we've only been able to find it at 2 populations. What we've learned from our surveys is that some of the cienagas are under threat from both hydrological and ecological change. Some of the hydrological changes that have happened in the last 30 years are head cutting and groundwater depletion. What we've learned is degradation of the watershed has probably contributed to degradation of the habitat for these orchids. Some of the ecological factors that we've noticed probably contributing to the decline in these populations for the orchid are encroachment of woody and shrubby vegetation into the cienagas themselves. So not only do the woody and shrubby vegetation take up water for other wetland plants, but they also shade out habitat for the orchid themselves. Some of the steps we're taking to improve the habitat is removing this woody vegetation. And we're working with collaborators to restore a natural fire regime to some of the cienagas. Fire was a natural part of these ecosystems over 100 years ago, but has been suppressed because of increasing human populations around these habitats. So not only will the fire reduce the populations of encroaching vegetation, but it should increase the groundwater levels, because that encroaching vegetation uses a lot of water that would be important in the habitat for the orchid. Cienega habitats are one of the rarest and most endangered habitats in Arizona. I'm drawn to working in cienega habitats, one, because they are so rare, but two, because they contrast so sharply with the rest of the surrounding habitats. They're verdant and green even in the driest times of year.
Oh, she did find one. Good girl.